And apparently the high viral load due to this high excretion of these particles is the responsible factor finally to reaching the mammary gland to insert there in specific sites where after, particularly in lactation periods, the cells may convert into malignant tumors and form murine mammary cancers. If we immunosuppress these mice, then we act on these types of lymphocytes. We suppress the super antigen stimulated lymphocytes, the virus production, and with it the viral load is going down, and thus the risk for developing the respective form of cancer is decreased. Now, is there something similar in human breast cancer? I'm pointing this out only for the fact that we may look at the wrong target by analyzing human breast cancer if we suspect that infectious agents may play a role, because in this case, events may have happened already, but something else carried by the lymphatic system might be more responsible. I will discuss also some agents which come into questions later on, which could play a role in these cases. Let me come in the next step here to a picture which I've shown at many occasions, namely, can we link some infections which occur in our domestic animals to human cancer? And the reasoning behind it originates from the fact that we do know a number of human pathogenic viruses, particularly the polyoma type viruses, BK and JC, but also some human adenovirus types, to a more limited degree Epstein-Barr virus and high-risk human papilloma viruses, which upon transmission into species in which they cannot replicate, where they are replication incompetent, they cause tumors, some of them quite actively, like the JC virus, even gliomas in hamsters, as it was shown in the past. Now, in the human host, these viruses all replicate. They are replication competent here, but apparently only in very rare examples, like in those shown here, but so far not for those types of agents. They are not inducing cancers in humans. Now, is it possible that some of our domestic animals harbor similar viruses which replicate in these animals, are excreted for a lifetime of these species, and yet do not form tumors, but upon transmission to another species in which they cannot replicate, that they become tumorigenic? That is the rationale behind the ideas which I